Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and we are revising chapter number 10, cell cycle and cell division. In our previous session we were reflecting upon mitosis, which is a vegetative division or somatic division, and we understand that at the end of mitosis one mother cell having diploid number of chromosomes 2n undergoes mitotic division and forms two daughter cells with the same number of chromosomes, that is diploidicity is maintained. Whereas in meiosis, which is a reductional division, undergoes or gets completed in two big stages, that is meiosis 1 and meiosis 2. In meiosis 1, again the meiosis 1 gets completed with four subphases: prophase 1, metaphase 1, anaphase 1 and telophase 1. And particularly the meiotic one is called reduction division. The reason being that at the end of meiosis 1, one daughter cell having two sets of chromosomes, like for example in human cells such as gonads, like say testicular cell, a testicular cell will be having 46 chromosomes, as all of you know, two sets, that is 2n, will undergo meiotic 1 and form two daughter cells at the end of it, and the two daughter cells will be having exactly 50% reduction in number of chromosomes. So that means 46 becomes 23, we call it haploid number. So at the end of meiotic 1, since we are getting two daughter cells with haploid numbers, we call meiosis 1 as RD, reductional division. So this gets completed with again four subphases: prophase 1, metaphase 1, anaphase 1 and telophase 1. Again, prophase 1 is the longest duration one because of its 5 substages. Prophase 1 being the longest one has got 5 subphases: leptotene, zygotene, pachytene, diplotene and diakinesis. And during leptotene, of course, as you may be recollecting from your memory, that chromosomes becomes distinct and visible under microscope. Compaction of chromosomes, this is important, this is the characteristic of leptotene, continues throughout leptotene phase. And second, during zygotene stage, chromosomes start pairing together, synapses will toss. So pairing of homologous chromosomes. The paired chromosomes are called homologous chromosomes, and synaptimal complex formed by a pair of homologous chromosomes is called bivalent or tetrad. And the third stage, during pachytene stage, crossing over. This is the most important one, which will not happen in mitosis. Crossing over between non-sister chromatids of homologous chromosomes occurs for exchange of genetic materials, most important. This is the significance of, and that is the reason why meiosis leads to formation of varieties. The crossing over is enzyme mediated process which involves an enzyme called recombinase, and the diplotin is recognized by dissolution of synaptimal complex and tendency to separate of bivalent except at the site of crossing over. This forms X-like structure called chiasma or chiasmata. And diplotin, of course, is having this bivalents which repel each other, and the sites of crossing over are called chiasmata. Points strongly appeared, and this is in some of the vertebrates, particularly in the oocytes of some of the vertebrates, their diplotin stage can last for months or years. So that's why this is the longest subphase of prophase 1 again. And in the last one, diakinesis, it's marked by terminalization of chiasma. The nuclear membrane breaks and nucleolus disappears. In metaphase 1, as always we expect, arrangement of chromosomes at equator and maximum condensation of chromosomes take place. And as you are seeing here in metaphase 1, all the chromosomes are lying in the equator. See, short and stumpy chromosomes within the center. And in anaphase 1, you can see the movement of chromosomes. Homologous chromosomes are getting separated, but sister chromatids remain attached at center. So, movement of homologous chromosomes without centromere getting broken, and nuclei are going to be formed. And in the last phase, that is during telophase 1, nuclear membrane and nucleolus reappears and cytokinesis follows. This is called dyad of the cells. So the basically telophase 1 results in formation of two daughter cells with haploid number n number. So parental cell has got diploid number 2n. We start off with 2n and end with n, and that too two daughter cells. And since the 2n is becoming n, we call meiotic 1 as reduction division, RD. Meiotic 1 is RD, reduction division. And immediately this will be followed by cytokinesis, because the two daughter cells have to be get separated. So cytokinesis takes place, and immediately after that we go to meiosis 2. For all practical purposes, we can say meiotic 2 is exactly of like mitosis. So we can also call it as equational division, also called homotypic division. It is meant for maintaining that number, diploid number. So it is initiated immediately after cytokinesis before chromosome gets elongated. In prophase 2, nuclear membrane disappears, chromosomes become compact, and in metaphase 2, you can see here in metaphase 2 again chromosomes are lying in the center. So when you do practicals also, when you focus the slide under a microscope, the moment you see the chromosomes in the center of the plate, take it for granted the cell is in under metaphase. So metaphase, meta means middle, center. So the chromosomes lying in the center, and in chromosomes align at equator, and what is important, microtubules attach with the kinetochores of sister chromatids, and in anaphase 2 centromeres get split and the chromosomes move to opposite poles. So north pole, south pole movement of chromosomes. So if you are able to see movement of chromosomes, then you understand that the cell is undergoing anaphase. And then of course in telophase 2, it is again cytokinesis. Short and stumpy chromosomes again will become elongated. So almost chromatin reticulum will be formed, and the nuclear membrane reappears, and the centrioles disappear. Cytoplasmic fibers will be not be there. Cytoplasm gets formed, and the number like initially it is now in meiotic 2, the parental cell is having n number, that is 23, right? And at the end of the meiotic 2, two daughter cells are formed, and both the daughter cells are having n number only. That's how we said meiotic 2 is the replica of mitosis, because the chromosome number is maintained. And thanks for that, because of the maintenance of the number, we are able to maintain the species identity. So significance, meiosis forms gametes that are essentially for sexual reproduction. As I said, the sperm and ovum in case of animals, pollen grain and ovum in case of plants are formed because of meiosis. So thanks to meiosis. Crossing over introduces new recombination of traits. That's what we said, genome gets exchanged. Helps in maintenance of chromosome number of sexual reproduction organisms. Provides evidence of basic relationship among the organisms. So what is the difference between the two, mitosis and meiosis? Takes place in somatic cells or vegetative cells, and meiosis of course takes place in gonads, reproductive cells. Mitosis single division which produce two cells as we discussed, and in the case of meiosis it is four daughter cells. And haploid and diploid both kind of cells may undergo mitosis, whereas here only diploid cells undergo meiosis, only gonads will undergo. Crossing over is absent, crossing over is very much present, and thanks to crossing over, because of that chiasma formed, because of chiasma there is exchange of genes, and pairing of chromosome does not occur. Here homologous chromosomes always get paired. So keep on drawing diagrams, appreciate what is going on in the nature, and when you label each diagram, understand the significance of each labeling. Take care.